Okay. That's that. All right. And hopefully you are seeing chicken pox right now. Is that true? Yes, chicken pox. Okay. Looking at yes, it's for right now. All right. Starting over. Here we go. We can do this. We're back in action. Sweet. All right. Chicken pox. Varicella zoster is the chicken pox virus, which technically within the umbrella of some of the herpes viruses, a plethora of different types of herpes viruses. This is just one. All right. So it has the opportunity to typically, we think about play this when we're little, giving us chickenpox, and then also going dormant on some of our nerve cells, and potentially later in life flaring up in the form of shingles, which is a rash-like manifestation that's more localized, whereas the chickenpox is pretty disseminated, lost on your trunk, but your extremities as well. This image is an electron micrograph showing the chickenpox virus, the varicella zoster virus, and its envelope, so you can see some of the membranes that surround the copy of this virus. So when a virus gets in, we start thinking about kind of like a growth curve, really, but it's a multiplication curve. So in this case, we're looking at bacteriophage. But the way that they are infecting bacteria, replicating inside, like we said, the livestock with those lytic phage, as they get in, hijack the machinery, make more virus, package it up, and explode out, right? So this is the number of infectious virions, so virus particles, if you will, on the y-axis, or that time on the x, hours of time that we're looking at. So the initial inoculation, we've got an amount of virus that's added to maybe the bacterial broth. There's a period of time where they're basically just figuring out their surroundings. It's kind of like a lag period. We call it eclipse. And it basically is where those virus, those bacteriophage, are finding their new host, landing, and starting to get in. Right? So, looking at number of infections, technically the number of virions goes down because they're getting into bacterial cells. They get in and hijack, so we start going through the multiplication cycle of those bacteriophage, and this is where we see the burst. It's like kind of like exponential growth as we're moving up on, we're replicating very rapidly and exploding. So in the process of this happening, you've got more virus, more virions that in turn are infecting other bacteria. So the cycle just keeps progressing over the course of time. And we can hit a plateau or a peak up here where we can measure and quantify the burst size based off of the amount of infectious particles, virions, that are released for bacteria. We showed some stuff last time with some of these pictures, and you can see bacteria that have a handful of virus particles inside of them, or virions, right? You don't expect them to have millions, but maybe a half a dozen or maybe a dozen particles that are inside that in turn are released out. And we could try to quantify that. There are handfuls of ways that we think about trying to understand how virus manifests in cells. And we can think about it with bacteria, and we can also think about it with animal cells as well. So we've seen, I don't know if it was this exact same image, but something pretty similar to this at the start of this semester when we were talking about different types of ways that we deal with microbes and potential safety levels when we think about pathogens that are super serious, right? So Ebola virus is definitely one of those. You can see this, this would be a biosafety level 4 lab. The lab has negative pressure, so all of the air in the lab basically stays in. If a door opens, air doesn't go out, it's pulling air in and sucking it out. The same with the hood, where they're actually working with stuff. There's negative pressure to keep things contained, if you will. Right? Their suits are the opposite, and they are positive pressure. They're blown up. There's air going into. You don't want to hear their leak. Right? That's trouble, right? And in turn, there's multiple things as we move through here. You can see the three tapes of their gloves. You can kind of guarantee they're wearing more than one pair of gloves too. So sealed up, and they've got their own contained ventilation systems that are piping in air and again keeping them protected. So there we showed as we started this chapter the tobacco mosaic virus, and I mentioned the first research with that type of virus to see, and I described it as being filterable. This is not the filter that they use, but it's a similar sort of filtration apparatus that we still can use. So we just trying to purify some stuff. So there's all sorts of ways that we might filter things, and different types of pores or little holes in the filter, allowing some stuff to get through. Right? There are sorts of membrane filters. I talked about the one with the tobacco mosaic, with it being a porcelain filter. So small pores in this glass, allowing, or porcelain, allowing things to move through, but really like virus, right? So what we've got in this example is a couple of different filters. One filter with a pore size of 5 microns. That's a pretty good pore size. Bacteria, I already told you, are about a micron or two, and they're getting through, right? The filter size over here is 0.2 microns or 200 nanometers, much smaller than the size of the bacteria, right? But big enough for virus to get through, right? So in our syringes, there's a handful of different things, right? The red balls in here are red blood cells, right? The blue ones would be some bacteria, and the green guys, those are the virus, right? So eukaryotic cells, quite a bit larger than the prokaryotic cells, are red blood cells, about 10 times the size. They're too big to get through this point, or this 5 micron filter. So eukaryotic cells, or our red blood cells in this example, they get trapped in the filter, but the bacteria and virus, they can move through it, right? The next filter size is too small, and it traps the bacteria, the eukaryotic stuff, and only the virus is able to move through there. And so when this filtrates down below, that's what we've got captured, right? And potentially wanting to carry on and figure out exactly what we're really looking at, right? This is a process that we use in the C-phase course when we're trying to identify virus in soil samples. We collect soil from different places, wherever the students feel that seems like a good spot, right? They collect a little bit of soil, they bring it in, and we try to extract virus that's in that soil that's potentially targeting some type of bacteria. Sometimes it's a simple collection where we basically just sort of wash it and try to pull it off. And others is an enrichment, so we grow it up after we've done some filtration. We grow that filtrate up with some known bacteria, giving it an opportunity that if the virus is there, who it wants to infect is also there, and we can ramp it up. So we call that an enrichment, and we try to get more of it. We do both mechanisms trying to see, right? really only think a couple maybe that might be targeting this one type of bacteria that I'm really thinking for, right? So there could be all sorts of different little green guys in this mixture of stuff that is maybe targeting different types of bacteria. But if I enrich the one, right, I would expect their population to be quite a bit bigger, and then I'd be able to actually maybe test some of those guys, if you will, right? So this type of filtration, similar sort of an idea, not the same filters, but similar idea, like they first utilized to visualize the tobacco mosaic virus being transferred through the filtrate slurry of tobacco cells that they filter through there, and what's left in that juice was the virus, right? This is an electron micrograph showing us some bacteria that's captured on that membrane, stuff that didn't make it through. And the pore size is small enough that the virus can, but the bacteria can't. That's really what it's going to show us. So there's a couple of different ways that we might try to see the virus, if you will, and blow it up, right? You can only really see it through the electron microscope, but you can do some different types of cultivation that give us some hints, right? The first one is flat. These are different types of flat. This is tissue culture flat. They lay horizontally. They're all nice and clear. You can put them right on a dissecting microscope and look through there, and you can see the animal cells, and you can also see if those cells have been infected. You oftentimes see different types of changes in those cells, like maybe fusing together, and then now it looks like this gigantic multinucleated cell. We call it a fusion. That indicates differences in potential viral infection, right? The animal cells growing up in there, the fluid basically keeps those animal cells alive, and you can watch the progression of it and see whether there's infection or not. These next images are bacteriophage images. Right? We call this a path assay, right? And you've got a bunch of bacteria that has been spread out on this plate, but right before it gets spread out, a little bit of a virus sample gets added to a little bit of the bacteria that's been added to what we call top augers, the media that's on the plate, but it's a little bit less concentrated, right? And so it all gets mixed together, and we pour it on the top of the plate, so you get this little thin film of bacteria and auger and possible virus all added onto the top. And it's solidified, and then it incubates up, and the bacteria, they're growing on this surface, making the plate look pretty cloudy because they're all dispersed in that media, right? And if the virus is there, when it infects those bacterial cells and it starts killing them, that burst bit of that multiplication code, it causes those bacteria to die. And we call this a lawn of growth. And the little pits or divots in this lawn are plaques. And we can count, maybe not on this plate, but on these other plates, we can count up these specific holes or plaques and quantify. It's a titer. And if we know the dilution, if we know how we diluted this initial sample, we can go back and quantify and say there are however many plaque-forming units, however many virus per milliliter or per microliter, and we can start gauging how much we're really dealing with, right? We do this type of stuff in the three-phase course and try to get good amount, counting up stuff, and then we grow them up to a point that's more than this. Right? You can see little bits of bacteria, but lots of plaques everywhere, right? That means you've got boatloads of virus, and then we add some buffer to the top of it, and we harvest the virus off. And then we do another one of these assays to see how much did we really get, right? How many plaques are we making per, 10 microliter drop that we add to the top of it? Once we know that, and we get massive amounts, like billions of virus per milliliter, right? Once we're up in about the billion range, and some virus might be a little difficult to get there, but usually after a couple of rounds of growth like this, you got huge amounts. Then you've got enough to get electron microscopy images because you need high concentration. So you've got enough to do that. You've got enough to extract DNA. You've got enough to actually do some science with. Right? A single little plaque shows us they're there, but you can't really do much with just one of those. Right? But we can try to get them amplified, right, and get this type of stuff happening. So these are like we started showing that bacteriophage linked up to part of that cell. The T4 bacteriophage is those icosahedral kind of spaceship-looking ones that target coli. This is a similar picture. This is supposed to really be at the tail end of the chapter, but I pulled it back up here because I like this. This is a pretty cool looking plaque out there. So we can see the bacterial growth in the background, right, all of the cloudiness, and then these plaques, which are much larger than these, right? There's all sorts of variation with virus, some little plaques, some big plaques, some in between. Some of them are kind of cloudy in the middle, and that indicates maybe like the lysogeny, where they go in and they don't kill them immediately, they hang out for a bit. But given the conditions on the place, they potentially kick it in and you'd see some of that. So there is some differences in morphology with these plaques, some smaller and bigger ones, but I like this one because it really can show the clearing a lot better than those other ones that were just little pinpoints. So there's other ways besides like those cell cultures that we saw that we might use for cultivation, and in this case, some chicken eggs are utilized when we think about different viruses, animal viruses. Influenza virus, when we make vaccines against it, we grow it up in embryonated chicken eggs, living eggs, not going down to the supermarket and buying a dozen eggs, right? They're fertilized eggs. And there's multiple ways, kind of depends on really what you're targeting, what sorts of stuff that we may inoculate these eggs so into the, yolk sac to embryo or the outer membrane, and allowing the virus to replicate and then extracting it out of it. That's the idea. So here we can see some more. This is similar, like looking at those cell cultures, those flasks. We could see cells as we start looking through the microscope at these flasks and see some different things. We're not looking really at this with virus. That's not what we're talking about in this image. But we could use these cells and study stuff like that, and we could infect them and see differences with it. Right? That's really what we're kind of looking at. And there's different ways that we may have cells for culture. All right? Here we've got normal cells, individual cells isolated from some tissue, in this case it's lung tissue. We've got a primary cell culture. These cells start to grow, they start to replicate, and normal cells have contact inhibition. So when they touch one another, they quit growing, right? And in these situations, when we're dealing with normal cells, you have to subculture this. You have to take some of these cells from this primary culture and move them into a new class with new media so that they can keep growing. Because otherwise, once they've all touched and we're right there, we're not going to live much longer. But if we keep propagating them and giving them space to proliferate, we've got this ability to keep them alive. A continuous cell line is different. And in this case, it cells from cancerous tissue. And these cells, cancerous cells, there's a lot of stuff with them where, they don't behave like a normal cell should be behaving. They don't die. And contact inhibition, who cares? They keep growing, right? So you can get this mass of cells that start to accumulate. This is a continuous cell line. It kind of minimizes the need for all of this extra stuff, right? And it depends on the circumstances as far as what we might be studying. Sometimes we use a continuous, and sometimes we do primary focus, and we're looking at some of these things. This sort of depends, right? Uh, so, and I was like, I know there's got to be something about the HeLa cells. So here we could see some fluorescence microscopy HeLa cells. Their name is Henrietta Lack. She ultimately donated these cells, and it was ovarian cancer, if I remember right. I think that's what it was. And there's all sorts of potential problems, benefits to medicine for sure. But some of this stuff really, they harvested this with kind of without their knowledge and went about doing it. So, kind of problem, right? And I'm not going to go through any details with all of that stuff, but there are, handfuls of things to talk about in a bit about who we had a lot that goes through some of those details and really has kind of got whipped out in the big scheme of things. So this image just showing a couple of different types of virus. I mentioned some of the syncytia a second ago, right? And that's what we're looking at here. We've got multiple fusion big cells. This is a paramyxovirus. Pox viruses, we can see some different inclusions as we're looking at some of these cells that have been infected with it. Herpes viruses, I said there's a boatload of different types of herpes viruses. We'll get some of those details later on. So we can see here some different inclusions. And adenovirus, different tips with that. So wrapping it up, just a couple of slides left. There's a handful of ways that we might be trying to identify besides the filtering that we just looked at, whether it be bacterial-based stuff or animal stuff. We're going to talk about our immune response. We'll go through details with that stuff, thinking how our body recognizes and tries to defend, but knowing that we've got an opportunity to produce some antibodies against some foreign invaders, sets the stage for all sorts of different immunodiagnostic tests using possible antibodies or potential antigens that we've made antibodies against. For detection becomes an opportunity to see all sorts of different things and maybe identify them, right? This example here is one that sometimes seems a little bit difficult to understand. And part of me is like, it's also, although we're talking virus stuff, I feel like it kind of might be better to really focus on this stuff after we've thought about some immunology in the first place, if that's okay, right? So we've got a handful of different reactions happening here, two different things that we're really looking at, right? This would be on a microtiter plate, so a little plastic plate with a bunch of little wells in the plate, typically 96 wells, and they look like little test tubes that you do a whole bunch of different reactions. And with that, you could also do a bunch of dilutions, so you could get titers. We could quantify how much antigen or antibody are we really looking at in the process of these solutions, right? That's kind of above and beyond what we really want to think about, right? So this first one is a control where we've just taken these red cells and added them to the walls of the plate. And as they get in there and kind of settle down, they create a little button, a little just clump of red cells at the bottom of that cell, right? Here we've got virus added to some of these red cells, and the virus and the red cells start to interact, we get a clumping, a hemagglutination, because it's red cells, and that well of the plate looks very irregular and spread out, not a nice little button, right? And the next bit, this is really the test, right? We add what we added in B, the red cells and the virus, but we also add a serum sample from that patient, so antibody within that serum. And if they have antibody against the virus, that's really what we're testing for, is this present? If it is, it links up to the virus and doesn't give it the opportunity to cause those red cells to clump. And so now a positive looks like our control up above. We call it hemagglutination inhibition. We're blocking this reaction that the virus would be causing. Right? Seems a little bit backwards. Right? It looks like our negative control, which in turn means it's a positive. Right? Crazy. There are all sorts of other techniques that we can think about with recognizing antigens and antibodies, and we can use some enzymes linked to that. There are also potentials for like radioactive molecules added to these things. That was some of the first stuff. And then recognizing we could get color changes or even light changes with some luminescent types of things. A lot of the radio immunoassays with the radioactive substances have kind of gone away, and it's more enzyme based, although sometimes they still use that. But deep down, I feel like, if you can avoid radiation, you probably ought to, right? I think that's probably a good idea. So these assays, there's all sorts of things. It's similar like a pregnancy test. Here we're looking for different things as we're looking at antigens and antibody and potential virus. That's what we're talking about, right? This could be, although here it's mentioning stuff like maybe some serum. We could think about a saliva sample. We could think about this like an over-the-counter COVID test, right? Could be saliva. It could be swab in your nose, right? But either way, we could do some of these things, right? And lots of the COVID is swab in your nose. Dang it, right? You don't want to poke your brain, but you get it clear up in there, right? So all of these things, we've got antibody, antigen, and then the purple glob on this other antibody is an enzyme. It's going to give us some sort of reaction, a color change typically. And if you've got this sandwich together, you get a color change, and it shows that you've got whatever you're talking about. And if it doesn't, if there's no antigen, this antibody can't link up to it, and it all rinses off. And then all you really see is the control that lights up, and the test sample doesn't, because none of this stuff links up in the first place. And there are boatloads of different assays developed for that. So shifting over a little bit, right? These are different, right? This is potato spindle tuber viroid. It is virus-like, right? It's a little different. And this is something, so we are in Idaho, but maybe you don't know all of the details about potatoes. You should, maybe you don't care, right? We grow potatoes with potatoes, right? To plant potatoes, you cut a potato and you plant that in the ground, and then it grows up the plant, and that plant produces a handful of potatoes underground, and we dig those suckers up, right? That's what happens, right? I remember being a little kid. I grew up on a potato farm, right? I remember being a little kid at elementary school. We get a new teacher. I can't remember where she came from, but in Pocatello, I don't even remember if I've ever seen a potato seller around here. You got to drive out a little bit, and then you'll see some potato sellers. But where I was at, she shows up and she's like, "What are all of these things?" She thought we were a bunch of wackos, and these were a bunch of bomb shelters. that we had built everywhere out in the middle of the country, right? There's all of these crazy bomb shelters. And I'm like, I don't think that's going to withstand a bomb, right? Really sure not. It's dirt and straw, right? Uh, oh, there are some other ones, right? So this happens at planting time, right? And you've got all of the potatoes, the sweet potatoes that you're going to be planting, and they get cut. The blades can potentially get contaminated if you got a potato that's infected and you cut it. Every other potato that you cut ultimately becomes infected. And then when they grow up and produce their progeny, their potatoes, they all look nasty, right? And in turn, not so palatable, right? I assume still probably edible, probably something that turns into potato chips because at the supermarket you're going to say, I'm not buying that, right? Uh, there you go, right? There's a little bit of agriculture for you. Okay, now, as we are wrapping this up, we can't talk about acellular stuff without dropping some mad cow disease into the mix, right? Prions are more than mad cows, right? Prions are proteins that are crazy, right? There are all sorts of different potentials for things to happen to stimulate this circumstance. And what it causes, when these proteins get stimulated, if you will, it causes uh, fungiform encephalopathy. It basically takes the gray matter and causes all sorts of porosity, spongy looking brain matter, right? Not what you want going on upstairs, right? So we've got these proteins normally in our body, the endogenous proteins, and the proteins are folded up and they function properly and they get altered or denatured and they change their shape, right? And that in turn causes other potential problems or dysfunction, right? So it could be a normal mutation that causes a misfolding. It could be an interaction where the normal protein comes in contact with a misfolded protein and it causes the spontaneous change in the green guy to turn into a red guy. Either way, we can see these proteins getting misfolded and causing these issues, right? And there are numerous potentials. It can happen, like I said, spontaneously. There are a couple of different types of human instances when we think about Jacob disease. Hulu was another one that was more of cannibalism. Foray tribe in New Guinea were consuming their dead loved ones so that they would live on within all of them. And those that consumed the brain, in turn, later on developed similar issues, and then they got consumed, and the cycle just keeps going, right? Typically with these things, and it depends on the circumstances, incubatory periods can be very long, right? Pushing decades potentially for these things after coming in contact. There are other opportunities where it's more acute. There were some outbreaks in the 90s in England where lots of livestock with mad cow disease had to be slaughtered, and the people that had become infected, and the progression of that was really pretty rapid, right? Kind of depends. So think about all sorts of possibilities with this. Here we could see a normal and an infected brain, the first Jacob disease, again, human manifestation with this, right? And that's what we're looking at with it. So the consumption of neural tissue has the opportunity to potentially infect, whether we're talking us eating other animals' brains, us eating human brains. There are potentials. I pretty much think that should just be off the menu, right? That would be better. So, that's it. And there are differences when we think about different types of animals. Scrapie is one that we see in sheep, the mad cow in cattle. There are others like chronic wasting disease that we see like in the deer and elk. I saw something in the news before class that was talking about a handful of deer that they're thinking about needing to euthanize to hopefully drive a wedge in there. In those circumstances, with the chronic wasting disease, it's not the ingestion of brain tissue, it's saliva that's able to transmit it. So it's a little bit different, right? And I will leave you with what I heard of this maybe last summer, maybe it was 2 summers back. There was a guy that was eating and eating squirrels because they're delicious, I guess, right? And apparently he was eating the brain, which I think must have kind of a nutty flavor, right? So he's eating these squirrel brains, and he ends up developing mad squirrel, I guess, and he dies from that similar sort of situation. All right, farewell, good day, happy weekend.